Hey guys, welcome back to One Stop Biology. So guys, today we are going to cover the genetic code. So basically we are now moving towards translation. And before we study translation, which is basically, coding for the amino acids or proteins, as we say, from the RNA, right? So before we do that, we'll have to understand how does the coding work exactly? Because till now we have seen complementarity and DNA to RNA, the bases are same, but it's not the same case in translation where you have RNA which is translated to amino acid sequences or polypeptides or proteins, whatever you can say it. So before we start genetic code, guys, please do not forget to like this video. Please do not forget to share it with your friends. And yes, your feedback is very, very important to me. I want to improvise more on the videos as per your, request. And please do not forget to give your comments. Either you like the video or if you have any doubt. So guys, let's start with the genetic code. So basically, during replication and transcription, what we saw that one nucleic acid was copied to form another nucleic acid, right? And I will again draw the same central dogma where you have DNA. It replicates itself. So replication is happening. Then RNA and then protein. Now here we have transcription and here we have translation, right? So now when replication or transcription is happening, what is happening is that one nucleic acid is getting copied to form another nucleic acid, right? So here what is basically happening is that it's complementary, right? But in the process of translation, what happens is that you are transferring genetic information from a polymer of nucleotide from a polynucleotide sequence to synthesize a polymer of amino acid, right? So basically what you're doing is that you are transferring genetic information from a polynucleotide to a polypeptide, basically, right? So here neither do they have complementarity, nor there is any relationship between nucleotides and amino acids, right? So basically there would be something which would be coding, say, from a particular nucleic acid, from a particular nucleotide to a particular amino acid, right? So basically, we'll study that itself in this video. Like if you see the biochemical nature of genetic material, and if you see the structure of DNA, basically genetic code deciphering the code can be very challenging, right? So basically if required, what happens is you will have to study a lot of things along with genetics, right? So basically it was George Gamow who was a physicist. Now see, he's not a biologist, he was a physicist who basically argued that since there are only four bases, we have four nucleotide bases, right? We have four nucleotide bases in DNA or RNA, right? So since there are only four nucleotide bases, and if they have to code for 20 amino acids, we have 20 amino acids, right? Then the code should constitute a combination of bases. Now there are just four nucleotide bases, but they have to code for 20 amino acids. So there should be a combination of these nucleotide bases, right? Now, basically, what he suggested that in order to code for all the 20 amino acids, the code, the genetic code should be made up of three nucleotides, is what he suggested, right? Now, this was, guys, a very bold proposition because, what he did is basically permutation combination. So if you basically use permutation combination for to the power 3 would be basically 4 into 4 into 4, which is 64 codons. Now we just have 20 amino acids, right? So basically, if it is coding, basically, if three nucleotides together are coding one base, then it would be then 64 amino acids, right? But that is not the case. We just have 20 amino acids, right? Now, but his hypothesis was actually correct, right? So again, but the problem was that proving his point was a very difficult task. So what happened is that by a scientist with the name of Hargobind Khurana, a chemical method was given, right? And basically, what he did is he again proved the same thing, right? Now, again, many scientists after him, including Marshall Nirenberg's cell-free system of protein synthesis, again helped in deciphering the code. Then we have different other scientists who said that yes, what initially George Gamow said was correct, right? So finally, the codons that we got. Finally, what we got? A checkerboard was given for the genetic code. Now, this is how this checkerboard looks like. Just see this, guys, once. This is how the checkerboard looks like, right? So, if you see the checkerboard here, now you will understand the salient feature of genetic code as well. Now, remember that these three-digit codes are known as codons, right? So these are codons of various amino acids, right? So see now what the first point is, the first feature of codons or genetic code is that codon is a triplet containing three nucleotide bases. One, two, three, right? So every time you see this, you will see three nucleotide bases coding for one amino acid. Now, by that method, we have 61 codons coding for 20 amino acids, right? So, we have 61 codons coding for amino acids, and then we have three codons. Basically, these codons do not code for any amino acid. These function as stop codon. These function as stop codon, right? Now then, some amino acids, right? So basically, this is the first feature. Second is that some amino acids are coded by more than one codon. Some amino acids are coded by more than one codon. Now, these kind of amino acids are known as, so basically the code is degenerate, right? The code is degenerate. Now, what is the third one? Now, the code is universal. Code is almost universal. So, say for example, from bacteria to humans to any other living organism that you can think of, you would always code for phenylalanine, which is an amino acid. Phenylalanine, right? So be it any living organism, triple U will always code for phenylalanine, right? Now there can be some exceptions in case of some protozoans, remember, right? Now, what is the fourth feature? The code AUG. Remember, code AUG, which is this here. It has dual functions. It has dual functions. It codes for methionine and it is also an initiator codon. AUG has dual function. It codes for methionine and is initiator codon. Remember, right? Now, fifth is what are the three stop codons? What are the three stop codons? Three stop codons are UAA, UAG, and UGA. So the three stop codons are UGA. So here you see this is the first, UAA, this is the second, and UAG. So we have one initiator codon, guys, which is AUG, but it also codes for methionine amino acid. And then we have three stop codons UAA, UAG, and UGA, right? So this basically is the genetic code. Now, what I have done, basically what this table is exactly, if I have to explain you, you know, how we have combined. So we need three nucleotide bases. So this is the first, second, and third. What we have done is in for a single box we have kept one UCAG. In the same way we have kept one UCAG. And here in the same sequence for one box we have kept all four nucleotide bases in the same sequence. So this basically is U from here, U from here, and U from here. The second one is U from here, U from here, and C from here. Third one is U from here, U from here, A from here. Fourth one is U from here, U from here, and G from here. This is how we get these four codes. And the first two basically say code for phenylalanine, right? So in the same way, we have got codons for all 20 amino acids, right? Now let's see how, mutation and genetic code are related, which is the next topic, that how mutation and genetic coding or genetic codes are related. So basically, guys, what happens is that the relationship we studied initially as well in a very short or crisp way, that mutations occur when genes kind of get deleted, gets inserted, or things like that happens, right? Now, sometimes what happens is that, large deletions and rearrangements in a segment of DNA happens. Now, this large deletion may basically result in loss or gain of a gene itself, right? And that is how a complete function can be added or can be deleted. Right. So let's take some example. So let's take a example of point mutation. Where what happens is that change of single base pair in the gene happens. Now, if we studied sickle cell anemia as well, guys, right? So in sickle cell anemia, what used to happen is that either in the beta globin chain or in the alpha globin chain, the single base pair would be changing, and because of that the amino acid residue glutamate which was glutamate would change to valine, right? Now this basically results in sickle cell anemia itself. This small change results in sickle cell anemia, right? Now you can imagine that the effect of point mutation that inserts or deletes a base can be very big, right? Now let's take an example as well. So say that three we know that three codes basically code for a codon, right? Now let's say I have a statement this, right? Now if we insert say a letter B in between has and red, then what would happen is Ram, and I said in between has and red. So this would be has. Now this would be BRE, then DCA, and then P. This is addition of B, right? Now say if we insert two letters, it would be Ram has. Now it would be BIREDCA and P. Now basically I'm inserting two letters. Now say I insert three letters. Ram has BIGREDCAP. Right? Now, basically, if we read these two statements, it's changed, right? Its meaning has changed. Here, Ram has a red cap. Here, he has a big red cap. The same thing happens in mutation when you have insertion. In the same way, we can even see deletions as well. Now, say if I keep on deleting things now, if I delete R first, it will be Ram has EDCA. So basically I've deleted R. Now I'll delete RE as well. It will be Ram has DCAP. I'll delete red as well. Then Ram has cap. Again, if you see this and this, it's changed. So whether you insert or you delete the base pairs, you will find that there is a change in the meaning, there is a change in the genetic code, and that is how we have a lot of disorders happening, right? So if you see that if basically insertion or deletion is completely changing the reading frame and we are getting a completely different meaning. Now this type of mutation, guys, this type of mutation is known as frame shift mutation. This type of mutation is known as frame shift mutation. So basically, this is frame shift insertion, and this is frame shift deletion, right? So now let's go towards the transfer RNA molecule one. So in the last video, I hope you would have gone through the last video. If not, please go through it. We studied that there are three types of RNA, right? And in today's video, we are going to study transfer RNA because it will be used in the next video. It will be used in translation, right? So let's see what exactly transfer RNA is. Now, guys, transfer RNA is the adapter molecule, right? Now, basically what it does. So what happened is basically, from the very beginning of the proposition of this entire genetic code, it was very clear to Francis Crick that there has to be a mechanism to read the code. It cannot happen that these codes are getting generated and it's not being read. It's not being understood basically, right? So basically there should be a mechanism to read the code as well as to link it to the amino acids, because amino acid themselves have no structural specialities to read these codes, right? So what is exactly reading the code then, right? So he postulated that there is a presence of an adapter molecule, right? Now this adapter molecule, so basically he said that there is an adapter molecule. Now, what is the function of this adapter molecule? So, this adapter molecule would first read the code, read the code, and would bind this code, would bind basically to a specific amino acid, would also bind to a specific amino acid. And at that time, it was given the name of sRNA, which is soluble RNA, which is soluble RNA, right? Now, these are afterwards known as tRNA, which is transfer RNA. So that is how transfer RNA was assigned the role of an adapter molecule. Now, how exactly it happens? How do we say that tRNA is the adapter molecule? So this is the structure of a tRNA, right? So basically this is tRNA. This as well is another transfer RNA. So this entire structure that you see, guys, this entire structure is a transfer RNA molecule, right? Now, what is happening here basically is that they have two ends. Remember that they have two ends. So see, guys, say if this is the 5:3 end, right? Again, basically this will be the 5 end. Right. Now, what is happening here is basically here you have it is attached to an amino acid. This is an amino acid. This as well is an amino acid. This is tryptophan, this is serine, right? These two are amino acids. So, what is said that it is bind to a specific amino acid. That is one, and it reads the code. Now, this is the codon. This sequence is the codon. Say here the sequence is A, G, U. And here it is UAC. Now what it is doing is this is basically an RNA. It is now translating, right? So this transfer RNA is getting bind here to read the code. Basically, it is reading the code here. So what it is doing is it is basically just reading the opposite nucleotide base. So opposite to A will be U, G will be C, and U will be A. Here again it will be A, U, and G, right? Now let's see what does UCA and AUG codes for, right? So basically what is happening is that if we see the entire structure, tRNA has an anti-codon loop, right? Now this is the anti-codon loop. Now that has bases complementary to the genetic code. So these are the bases which are complementary to the genetic code, and it has an amino acid acceptor end. So this basically is the amino acid acceptor end. Now to this amino acid acceptor at end, the tRNA binds to amino acids. Now, transfer RNAs or tRNAs are specific for each amino acid, guys. Remember, transfer RNAs are specific to each amino acid. Now, for any kind of initiation, there is another specific tRNA which is referred to as initiator transfer RNA, right? But there are no stop transfer RNAs. There are no tRNAs to code for stop codons, right? Now here you basically are looking at the entire tRNA molecule, which is coding for the amino acid. Now also guys, this entire structure is known as a clover leaf structure. This is known as clover leaf structure. So guys, this is the exact structure of tRNA, and this is how the tRNA acts as at the adapter molecule for the translation of amino acids from messenger RNAs or from RNAs. So, guys, we have finished the entire concept of genetic code, and in the next video, we'll start with translation. Again, a very, very important topic for your boards as well as NEET UG preparation. So, I hope you understood the entire concept of genetic code, and I hope you understood, how it causes mutation, how important the accuracy, is here, and how transfer RNA plays a very important role in translation. So, guys, please do like the video, please share it with your friends, and if you are new to the channel, please do not forget to subscribe to the channel to get all the updates. And yes, if you have any doubt, please let me know in the comment section. You can also ask your doubts on WhatsApp, and the WhatsApp number is given in the description of the video. But yes, guys, your feedback is very, very important. So please do not forget to comment on the video whether you liked the video or you have any doubt. Thanks, guys. Bye bye.